Presented by Caltech.
Okay, well, good evening and welcome to tonight's Ernest C. Watson lecture. I'm Doug Reese, Professor of Chemistry in the Division of Chemistry and Chemical Engineering. Before introducing tonight's speaker, I'd like to remind you of the next Watson lecture that will be on January 21st, featuring Julia Greer, Professor of Material Science and Mechanics, who will be speaking on Size Matters, the importance of building small things. Now, it is a great pleasure to introduce my colleague, Professor William Clemens, as tonight's Watson lecturer. Bill has been at Caltech since 2005 and is currently Professor of Biochemistry in the Division of Chemistry and Chemical Engineering. And he's also the option rep for the Biochemistry and Molecular Biophysics graduate option. Academic life is an itinerant one, and that's doubly true for Bill, who grew up in a military family, as I think you'll be able to tell by his Marine-style haircut. Bill received his undergraduate degree. The short story for his growing up is that he was born in Virginia, migrated to Hawaii, and then returned to Virginia, where he finished high school and went to college at Virginia Tech. For his graduate work, Bill went to the University of Utah, where he worked with Professor Venki Ramakrishnan. For his thesis, Bill was instrumental in determining the crystal structure of the entire 30S ribosomal subunit. This project represented a new and highly risky direction for Venki, and Bill was the first student to lead the charge in solving what originally seemed like an impossibly challenging project. That it worked out spectacularly well is really no understatement, as it culminated in Venki sharing the 2009 Nobel Prize in Chemistry. In Venki's Nobel Prize biographical, he writes, "Bill Clemens was a smart and very ambitious student who fortunately was at a stage where he was willing to gamble on something potentially big. His eyes lit up at my suggestion, and he immediately said he would take on the project." So this drive to gamble on big, challenging problems is really a fundamental characteristic of Bill and is clearly reflected in tonight's Watson lecture. Bill's work continues the exceptional tradition of crystallography and structural biology that originated with Linus Pauling at Caltech and continues, I think, as one of the traditional strengths here. I should add that this area will be featured in an article appearing in the March Engineering and Science, including discussing Bill's work and other work here in this area. Bill's research targets poorly defined biological systems for mechanistic analysis using a structure-based multidisciplinary attack. His studies have involved the most challenging types of biological problems: membrane proteins, also known as Greasy proteins, which will be feature in the title, complexes and complexes of membrane proteins, and his outside-the-box thinking about these developments, or these problems, has been recognized by an NIH Director's Pioneer Award. Bill will share these developments with us tonight in his lecture, How Do You Make a Greasy Protein? Bill?
All right. Okay, so thank you, Doug. It's really great to be able to present the work that my group has been doing, and this is an intimidating audience, I hope you know. So I do feel some pressure to do a good job of representing our science and the key people in my group. So the story I'm going to tell you today is one that really is based on this problem of understanding greasy proteins. And so that's the fun science title of my talk, How Do You Make Greasy Proteins? And I have to admit, when I came up with that title, I was trying to think of a way to represent membrane proteins that was conceptual until I discovered that my wife's colleague saw this title and went, "Oh, that sounds gross." So maybe I missed the mark, and so maybe we'll rephrase greasy as representative of a title that's more typical of a talk that I'd give, which is a molecular view of membrane protein biogenesis. And so what we mean by this is we're interested in membrane proteins, which are these greasy proteins, which you'll understand by the end of the talk. And these membrane proteins have to be created, right? And they're made in cells, and that's where the biogenesis component comes from. And myself and Doug and others here at Caltech are structural biologists, and so we actually want to understand these macromolecular structures at atomic resolution. So we want to understand them at detail, and at detail that allows us to gain mechanistic understanding. And so we look at this from at the molecular view. We want to get to atomic resolution to understand the molecule. And so nominally, the way to title this would be a molecular view of membrane protein biogenesis. So that said, that brings me to a kind of interesting figure, which is this figure here. So this picture is of the endoplasmic reticulum. This picture was taken, in the early 1950s by a person named George Pilate, and this picture really is representative of myself, the work that my lab has been doing. And so everything in my career, starting with the working on the ribosome and then trying to understand membrane protein biogenesis, is all wrapped up in this figure. And so this figure really kind of defines my work, and we're going to use this as a template for kind of understanding the problem. When these types of figures were made in the 1950s, and it really was born out of the fact that it was a new era when electron microscopes had been invented, and George Pilate turned out to be someone who actually was really good at making specimens that could be analyzed by electron microscopy. And so George Pilate, along with Albert Claude and Christian Dedeuve, basically developed our understanding, our subcellular understanding of cells, right? And they won the Nobel Prize in 1974 for this work, understanding the functional organization of the cell. And so, it's this work by Pilate that, on some level, he's actually a scientific grandfather of mine. And so understanding the molecular details of this has been a pursuit for myself and people like me for many years. Okay, so biological membranes are going to be the main topic of this talk, and many of you probably have some concept of that, but I want to make sure that we're all on the same page. So we're going to step back and do a little bit of a historical picture, or a historical trek through our understanding of biological membranes. What I've drawn here are two cartoon images of two typical cells. Okay, on one side is a bacteria. This is sort of a prototypical E. coli cell. It's made to scale relative to a eukaryotic cell, as you might find in a cell culture experiment. And of course, this is a cartoon that I just threw together, and so this is grossly oversimplified. But what you can see is that in both of these cases, we have a cell, and that cell encloses this blue space here. The blue space we refer to as the cytoplasm. This is the water environment that's inside the cell. And in both these cases, these cells are surrounded by bilayers, these biological membranes, and that concept will be more clear as we move forward. But the way to think about these biological membranes is that these biological membranes really are like water balloons. And so in a structure, in the cartoon form we have over here, this would be a water balloon that the membrane of the balloon itself basically defines the inside of the cell and the outside of the cell, and it makes a barrier preventing things from being able to go inside and out. Now, what you'll note if you look at the eukaryotic cell is that things get more complex in the bacterial case. In the bacterial case, it's just a single water balloon, and so lots of interesting things happen on the inside, but from our perspective for this talk, it's actually a very simple system. Whereas in eukaryotes, we actually get more complicated because inside that water balloon are a bunch of smaller water balloons that we call organelles. And so all of these other cartoons that I've drawn in here are different types of organelles. In green would be the nucleus, and pink would be the endoplasmic reticulum. These would be Golgi apparatus, these would be mitochondria. Very simplified. But the idea is that in all these cases, the dark band that surrounds all of these cases are biological membranes, right? And so clearly they play very important roles, and so we want to understand them in detail. And so what do we know? So if we're going to zoom in so focus on this eukaryotic cell, what we can say is that cells and organelles are surrounded by these impermeable membranes. All right, so the biological membranes intrinsically are designed to keep the outside out and the inside in. And if we want to get some scale for this, if we take this as a typical eukaryotic cell on average is about 20 microns in diameter, and the membrane itself is going to be about 5 nanometers, right, 0.005 microns. So relatively speaking, it's really small. And in fact, if we try to draw this to scale, what we'll find, so if you note going between the two figures, I'm going to reduce the thickness to a scale that's really more appropriate. And what we find is that these cells are surrounded by these extremely tiny membranes. And just to give perspective, if we take the Beckman Auditorium, right, this building that we're all in, and we define the outside of it as surrounded by this biological membrane, the membrane that surrounds this building would be about 1/4 of an inch thick. right? So they're extremely tiny, and yet they have this really important role because they have to provide and define this barrier that holds the cell together. And that being said, they also have to prevent things from being able to come in and come out. But that's a problem because obviously information, nutrients, and things of that sort have to be able to cross these bilayers. And that's where membrane proteins come in. So membrane proteins live in these bilayers and allow things to enter and exit through this membrane barrier. So membrane proteins, what are they? Well, the simple definition for membrane proteins are that they are proteins that are embedded in this lipid bilayer, this biological membrane. And the concept of bilayer will be clear by the time we're done. Membrane proteins are responsible for the trafficking of information and nutrients across these membranes. So all of the information that's to travel between these has to go through these membrane protein conduits. And because they play such an important fundamental role, about 20 to 30% of our genetic information is dedicated to making membrane proteins, right? So our genome defines about 20 to 30% of our genes are actually membrane protein genes. And so that's a huge number of genes. And just from that perspective, you consider it to be a very important problem to understand. But even perhaps more interesting, at least for those of us who occasionally have to take pharmaceutical products, about 60% of the approved drugs that are on the market target membrane proteins. And so understanding membrane protein structure and the mechanistic aspects of membrane proteins allows us to understand why those drugs work, how can we improve on those drugs, and how can we think about developing and creating new targets. Now, of course, all of that's really important, and so a lot of effort's been put into trying to understand membrane proteins. But the reality is even to date, we still know very little about how they work. And so labs like mine and like Doug's and like a lot of other labs throughout the country, throughout the world, are really trying to improve on this, right? We need to understand these molecules because they're important. And so structural biologists like myself have been putting a lot of effort into solving molecular structures of these molecules, which provide the blueprint for understanding how these proteins work. On the right here, we have a chart that really talks about structural biology from the perspective of where we are today and the success of structural biology. So some of you may remember the first structure was solved in 1957. That's the first protein structure by John Kendrew of myoglobin. And from then to now, we basically started in 1957 with the first structure. Now, if you go to the database where we store structural information, we have over 100,000 protein molecules whose structures that structural biologists like myself have been able to determine. For membrane proteins, however, the lag is about 30 years, and so it wasn't until the mid-1980s that the first membrane protein structure was solved. If we then scale it up to now, where clearly in understanding membrane proteins, there's a huge lag behind soluble proteins, and in fact, as of today, we only know about 500 unique membrane protein structures. Well, that being said, that's still an amazing amount of information, so we've been doing a good job with what we've got. But it reflects this problem with membrane proteins that making membrane proteins is hard. It's hard for the cell, and it's hard for us to actually reproduce and do it in a way where we can get enough protein to do something interesting. Okay, so understanding membrane proteins structurally is important, but also trying to understand how biology makes membrane proteins is really important as well. And the reality is that all membrane proteins, and in fact secreted proteins like insulin, anything that has to be secreted outside the cell, starts here in the endoplasmic reticulum. And just to kind of relate this picture to this picture, the endoplasmic reticulum is this pink bits here, right? And so this endoplasmic reticulum is this network of membranes where we actually use this for secretion and for making of membrane proteins. And so let me orient you just to give you a little bit more picture of what we're talking about here. So you look at a picture, and it's sort of hard to tell which parts of the endoplasmic reticulum here. If we zoom in, I've now colored in, so between these dark lines here, that's actually the endoplasmic reticulum itself, okay? So that's the organelle that we're talking about there. And if that defines the inside, if that defines the organelle, this endoplasmic reticulum resides in the cytoplasm. I should add, the cytoplasm is where proteins are made, and that'll come back in a minute. But if the endoplasmic reticulum is here, then the space outside of that is the cytoplasm. It defines the space here. The space within it, just for nomenclature purposes, is called the lumen, so the endoplasmic reticulum, which I'll default to the ER, is this space between here, right? So the lumen fills the space between these bits here, and then these black lines are, again, membranes, okay? And in this particular case, these are ER membranes. So these membranes, like all biological membranes, are made up of molecules that are called lipids, and they look something like this. And so that's just to give you a frame of reference. We're going to come back and understand why that's a really cool molecule and what these kinds of molecules can do. Okay, so if we want to understand the details of what's going on in this membrane here, we really need to go back fairly early, and in fact, back about 2,000 years in our understanding of surface chemistry. Because really the first sort of interesting historical notation where we began to understand the modern concept of membrane proteins started with Pliny the Elder. So Pliny the Elder, operated about 2,000 years ago. He was a Roman, and he's credited with writing the first encyclopedia, right? So he was aggregating all of this knowledge, and one of the things that he wrote, a story that he related in this encyclopedia, was about was a story about sailors working in ports or on the ocean. Sometimes the water would be churned up by the wind, and they would pour oil on the water, and that oil would have the effect of actually smoothing that water. Right? And so he noted that they would use oil to actually flatten or still the waves, to tame the waves. So the next place where we actually have kind of a historical record that relates to the story we're telling comes from Ben Franklin. And I should add, the names that I'm throwing up here are just amazing people historically. I mean, Ben Franklin, no one thinks of him as a surface chemist, but he actually did the first surface chemistry experiment that was documented. And so Ben Franklin was on a voyage from England to the U.S., and he was there were a bunch of other boats on this voyage with him, and he noted that while they were sailing along, all of the boats were making wake, except for two. Two of these boats didn't have any wake behind them, and being a scientist like he wanted to know why. Why was that happening? That was weird. He couldn't explain it, and so he went to the captain, who, in his own words, or in Franklin's own words, described very angrily or annoyedly, well, of course, that's the cooks have just dumped their waste into the water, right? And so Franklin, being the scientist that he was, connected these dots where he'd read Pliny the Elder as a young man, or in his youth, he says. And he said, wait a minute, Pliny the Elder described this story where they put oil to still the waves, and this captain says that they're dumping their food waste into the water. That's stilling the waves. Well, food waste is full of oil. And so that must be what's happening. They're stilling the waves. And so he decided that he wanted to test this experimentally. And so that takes us to something that is generally referred to as the Ben Franklin experiment relative to surface chemistry. And so what Ben Franklin wanted to do was he wanted to do this in a more controlled environment. And so I'm going to actually read this to you. This is his account of the first experiment that he did, which is from a letter that he wrote to William Brownrigg, who was a member of the Royal Society in London. And basically he says, at length being at Clapham, where there is on the common a very large pond, which I observed to be one day very rough with the wind. I fetched out a cruet of oil, a little jar of oil, and dropped a little of it on the water. I saw it spread itself with surprising swiftness upon the surface. And there, the oil, though not more than a teaspoonful, produced an instant calm over a space several yards square, which spread amazingly and extended itself gradually till it reached the lee side, making it all that quarter of the pond, perhaps a half an acre, as smooth as a looking glass. Right, now that's hard to believe that experiment actually works, but it did, right? He spread oil, and he saw that a teaspoonful of oil spread more than half an acre. So that's amazing. And the thing about that experiment is that Franklin, with that experiment and a series of other experiments, recognized a couple of things. One, that oil was spreading over that entire surface and was spreading extremely thinly. The other thing that he noted was that the oil would push things out of the way as it was spreading. And so there was some surface tension that was happening there, and there were forces that were at play. And so he made a bunch of connections that were just really amazing with such simple experiments. The next players historically in this really built off that Franklin experiment, and this would be Lord Rayleigh, or John William Strutt, who had a lot of important scientific discoveries. Notably, he won the Nobel for discovering discovering argon. But in addition to this, he did some surface chemistry experiments, and those surface chemistry experiments really actually got us the first scale of molecules, and that will make more sense in a minute. Interestingly, after he published that initial work, a young woman named Agnes Pockels, she was a German, and she had finished high school and gotten a decent high school education, but at the time was prohibited from furthering her studies because of her gender. And so she was forced to actually pursue her further studies on her own, and she would get her brothers to bring her papers, and she'd read vociferously. And she was struck by this experiment that Lord Rayleigh had done, and actually in her own kitchen improved on his experiment and actually got a better number than Rayleigh did in terms of understanding the scale of a molecule. And so Lord Rayleigh actually got us the first scale of a molecule. The final sort of person from the historical perspective of the Ben Franklin experiment is Irving Langmuir. Irving Langmuir is really probably recognized as one of the most famous surface chemists. He's the person who actually came up with molecular models, and so Langmuir took these same exact kind of studies and improved upon them to the point where he can make very accurate measurements. And for that work, he actually won the Nobel Prize in Chemistry. He worked for General Electric at the GE labs, and what was interesting about him is this was one of his many projects. So he's famous actually for vacuum tubes and many other different types of work, but like improving light bulbs and these sorts of things. But this was something that, a scientist like me, that's what he's remembered for. All right, that's great. But I think it's kind of cool, you look at this historically, in these very simple experiments, you can learn some really amazing things. And so let's do that experiment ourselves. And so, we're going to do the Ben Franklin experiment. And this is a graduate student of mine, Ji Young Mok. And so Ji Young's going to actually run us through this type of experiment that Ben Franklin did. And so on one hand, she has just a simple powder. On the other hand, she has a bottle of olive oil. And what we're going to do is we're going to take a 1 by 1 meter square of water, and we're going to cover that 1 by 1 meter square with this powder that she has. It's like a podium powder. It's not really important what it is, but we need the powder there to actually be able to see the surface properties in action. Unfortunately, our first experiment, we used too much powder, so we reset and started over again. And now what she's going to do is she's going to put 1.5 microliters of water on the oil, and we're going to watch what happens. And what you see, just like Ben Franklin saw, is that tiny little amount of water spreads a huge distance, right? So that 1.5 microliters goes from that 1.5 microliters, which is really hard to relate to you here, but that's 1,000th of a mil, right? It's a tiny amount of oil, and yet it spread, and we actually measured this, it spread 55 centimeters. Other things that are sort of interesting when you take an experiment like this is that tiny bit of oil had to spread an amazing amount, but it also had to push all that powder out of the way as well, just like Franklin noted, that it's actually exerting pressure around it, and that pressure is going off in all directions. All right, well, that's the experiment. What can it teach us? And so as Lord Rayleigh noted, we actually learn how big a molecule is from an experiment like that. And this turns out to be really simple, right? We know we have 1.5 microliters of water. We know it spread 55 centimeters diameter for a circle. And so all we have to do is just we just can use the equation for calculating the volume of a cylinder, albeit a very thin cylinder, and use that to calculate the height of the water. And so we just rewrite the equation like this, and then we plug in our numbers. We know the volume, we know the radius, and if we do the math, what we get the height of that thin layer as it's spread is 6 nanometers, or 6 times 10 to the 9th meters. Right? I mean, it's just really hard to appreciate that number, but that's extremely tiny, right? And what we can take from that, and what Lord Rayleigh took from that, was that this oil that was spreading, this olive oil, as it was spreading, the molecules that compose this couldn't be bigger than 6 nanometers. Right, and so that's our number, but he got a relatively similar number, and so we know that we can measure the size of a molecule based on this. It's just an amazing result from such a simple experiment. Now, as I said, there is a molecular model that underlies this. This is what Langmuir discovered, and did a lot of really great things with this. But the molecular model basically starts from this principle that olive oil itself is a mix of fatty acids, right? It's a mix of fatty acids, and these fatty acids have mostly hydrophobic components. And so for olive oil, the main fatty acid in olive oil is called oleic acid. And oleic acid looks like this. It's got 2 oxygens on one end, and the rest of it's all carbons. These 2 oxygens are happy to interact with water, but the rest of these carbons here want to repel water, right? They're hydrophobic. So you've got a hydrophilic part and a hydrophobic part. And if you take this molecule and you put it in water, what this molecule is going to want to do is want to get these hydrophobic bits out of the water. And so what happens is this fatty acid will reorient itself to actually stick its hydrophobic tail out of the water into the air. The air is a reasonably hydrophobic environment for this context. All right, and so if that's what's happening, if we add the millions of other molecules that are present in, perhaps billions of molecules that are present inside that 1.5 microliter drop, what we see is that those molecules are assembling themselves to form this very simple monolayer, right? They're spreading to form this layer 1 molecule thick, right? And so that measurement that we made really is telling us exactly how thick this monolayer is. All right, so the question then is, how did we do? In our experiment, so we measured about 6 nanometers based on our work. And the reality, the size of this is probably more in the order of 3 nanometers. So we're off by an order of maybe 2 or 3 times. But yeah, it's pretty close, I think, considering how cheaply that experiment was done. And so if you do it more carefully, as Lord Rayleigh did it, you'd get a better number. All right, so in fact, Langmuir did it much more carefully and got much better numbers. But what Langmuir also, it was just amazing what he could do with this type of experiment, because he could measure the forces and he could actually understand not only that this was one molecule thick, not only how these molecules were oriented on the surface, but he actually was able to determine there was a kink in this molecule. So they didn't know the structure of the molecule, and he could figure out that there was a kink in it based on these kind of very simple types of experiments. All right, so we know that if you have something like a fatty acid, it will rearrange itself in a water environment to keep the hydrophobic bits out of water. All right, but that doesn't tell us anything directly about biological membranes. And so there's a few more historical notes that I'd like to point out. So cell membranes, biological membranes are composed of lipid bilayers as implied already. The first person really, I think, or the person who's credited most for our understanding that these are made of lipids was Ernest Overton. Ernest Overton was a British scientist. He was interested in anesthetics, and so he was studying anesthetics using tadpoles. And so what he would do is he had tadpoles in a jar, and he would add anesthetics to them, and he would try to observe what anesthetics would actually knock these tadpoles out. And the connection that he made was that the molecules, that the more the molecule that he added, the more soluble it was in oil, the faster it worked. Right? And the simple connection he made, which is just a big leap, was that the barriers in cells had to be composed of something that was similar to oil. Right? And so what that meant was that membranes have a greasy component of it, right? They're this oily component. Right? That's really cool, right? And so eventually it was realized that the main component of these are lipids. And so the next major experiment that was really credited with our modern understanding came from Everett Gorder. He was a pediatrician in Holland, and he, working with his student Grendel, decided that they wanted to try to understand this molecular mechanism for this as well. Doing very similar experiments, they basically took red blood cells. Red blood cells, they could easily, are always about the same size, and so they could take a whole bunch of them and measure the surface area of a red blood cell. And then he could extract the lipids and then measure using something just like Langmuir did. He can measure the volume of the lipids in that lipid bilayer. And then he could ask the question, what is the ratio between the surface ratio between the lipids themselves and the cell? And what he gets doing that experiment is a ratio of 2 to 1, right? And so that bilayer was made of two molecules thick. All right, and that was really a cool result, okay? And so these are made of lipid bilayers. Lipid bilayers are, let's see if it's going to transition. Lipid bilayers are made of lipids, already implied. Lipids, basically look like this. This is really an atomic representation of this. These spheres representing the sort of main orbital, or the main outer shell of the atoms themselves. We can make the picture a little more simpler by removing that kind of information and really just focusing on the bonds. And what we can see is that if we look at a lipid, so this is a stereotypical lipid, and what we can see is that there's two components to it. There's the head group. This head group is hydrophilic, right? It's got lots of atoms. It's got a phosphate. It's got oxygens. It's got a nitrogen. All of these things are happy to interact with water. On the other end, it's got fatty acids that are mostly which are almost completely made of these long acyl chains, these carbon chains, which are hydrophobic. And so the nature of it, just like the fatty acids by themselves, means you've got a large hydrophilic component and this very extended hydrophobic component. Okay, so we can take this structure, make it a little more simpler. So let's reduce it to an even more cartoon version of a lipid. And so we have a head group. We've got these acyl chains that are attached to them. And the beauty of these acyl chains, of this structure, is that if you actually, on a molecular level, these lipids actually are about the size of a cylinder. Okay, you can imagine them to be like cylinders where they have a hydrophilic top and the rest of it's hydrophobic. And that allows these lipids to actually assemble themselves together. Right? And they assemble themselves together in a lipid bilayer. So we saw a monolayer before. Because of the structure of these, they actually have the ability to form a bilayer. And so what you get is on the top, you've got all of these lipids which are associated side by side. You get the hydrophilic head groups on top, these interacting with water. And then below that, you have all of these acyl chains, which are these tails, which is the hydrophobic bit. That's one surface, and underneath it you have exactly the same surface. And those come together to protect this hydrophobic components of these molecules from the hydrophilic water environment. All right, so that's what these membranes are made of, these lipid bilayers. But the thing is that these lipid bilayers are great because they actually, the forces that drive them together are actually fairly strong forces, and they can work as really perfect barriers for cells. The problem is that things have to get across these, and most of the biological molecules are hydrophilic. They can't cross this. And so the next component of our understanding of biological membranes really came from John Singer. And so this is called the fluid mosaic model. And so they took that bilayer concept and actually using a lot of different experiments began to understand that that biological membranes aren't just composed of lipids, they also have proteins that are embedded in them. And these proteins are responsible for all of the information that gets transferred across. And the special thing about these proteins is that they have to live inside this hydrophobic environment. And so membrane proteins have this extremely hydrophobic surface. Now, we can actually simulate this in a computer, get something that looks like this. And so here we have a membrane protein. This is a structure that's solved in my group. We've actually embedded this computationally in the lipid bilayer. We're running a molecular dynamics simulation to kind of imitate what's actually happening in cells. And so what you can take from this, here's our lipids. And so this is our membrane protein here. These green things are alpha helices. That's the structure of which was first understood by Linus Pauling. But what we can see is this protein has 6 transmembrane helices. These transmembrane helices are hydrophobic, which allows them to actually exist inside this, in this bilayer. And the bilayer itself, you can see these acyl chains are assembled together. They're extremely hydrophobic. And because they're hydrophobic, they're held together. And on the other end of it, these head groups are bathed in these red circles here, which is just water molecules. Right, so a bilayer looks something like this, where you've got proteins in constant motion, but because of the bilayer, the thing, and this relates to this fluid mosaic model, is that the lipids are in constant motion, but because it's hydrophobic forces that are holding it together, they can move quite a lot, but they still maintain this rigid barrier. And so now, we know what's going on here, right? So these black lines represent membranes that are full of, that are lipids and protein, right? Now they're fluid mosaics, so they're in motion, but they actually have this very hydrophobic component that they consist of. Okay, so the other piece that we're missing in this picture are these little black dots here. These little black dots turn out to be ribosomes. Ribosomes are protein factories, and so ribosomes are the site of protein synthesis. So all proteins and all organisms on the planet are made by ribosomes. Ribosomes themselves are these amazing, huge RNA protein complexes. And so this is the structure of a bacterial ribosome here. As implied by Doug, the ribosomes come in a couple pieces. So this on the green would be a small subunit, the 30S subunit, and blue would be the large subunit, the 50S subunit, and this is what we worked on when I was a graduate student. But these are huge complexes that make proteins, and they basically take the genetic code of DNA and convert that, it gets converted into RNA, and then that RNA code gets converted into the amino acid code, which are proteins. They're just long polymers of amino acids, and that's encoded by nucleic acids. And ribosomes are responsible for doing that. So ribosomes are going to make proteins, and when they make these proteins are going to merge in a sequential fashion out of the ribosome, right? So you make one end of the protein, and as you add more and more, that protein is slowly being extruded from the ribosome. And eventually, for most proteins, the ribosome will make for most systems, the ribosome will make the protein. It will come out of the ribosome. It will go fold into its structure, and then go on to do what it wants to do. In the case of membrane proteins, however, we've got this problem because membrane proteins are extremely hydrophobic. Right? Membrane proteins, they have these hydrophobic surfaces, and so they can't just be simply pushed out into the environment. Because of their hydrophobic nature, they'd actually aggregate, right? They would assemble just like lipids do. Their hydrophobic bits will assemble together, and they would crash out of solution, and there's big problems. They don't get to do what they need to do. And so hydrophobic membrane proteins have this additional level of complexity in how they get made. And so they're going to be made by the ribosome, just like all other proteins. But the really sort of important understanding here came from this concept of the signal hypothesis. And this is the fact that all proteins that need to be targeted to membranes have a signal that's encoded in them. And the person who really came up with, who did some of the pioneering work and actually connected the dots in this was Günter Blobel at the Rockefeller. And what Günter Blobel postulated was that there was a specific signal sequence at the start of the protein that was responsible for targeting proteins to the endoplasmic reticulum. Very simple concept that proteins are flagged with a sequence at the start of the protein that says, deliver me to the ER. And he, with David Sabatini, came up with a cartoon to describe this. And so this is published in the early 1970s, and what you can see here is ribosomes assemble on a messenger RNA. The ribosome starts making the protein. There's a signal at the start of the protein that emerges from the ribosome after a certain amount of time. When that emerges, that tells this protein you need to go to a membrane, specifically the endoplasmic reticulum membrane. Other factors help this protein get into the membrane, and then either gets things secreted across, or membrane proteins will get inserted laterally into the plane of this bilayer. Well, okay, so it's interesting, this is a cartoon that eventually won Blobel the Nobel Prize as sort of an interesting sketch that would give you a Nobel Prize. But the thing that's also interesting about this is before the Singer fluid mosaic model. And in fact, they don't show any proteins embedded in the bilayers. They didn't even know that at this point in time. So, okay, so ribosomes make all proteins. So ribosomes make all proteins, right? And that includes membrane proteins. And for most membrane proteins, when they're being made on the ribosome, so here's our ribosome, here's a messenger RNA, proteins are being made, so the amino acid sequence is now coming out of the ribosome, and at the start of the protein, there's a signal, right? For membrane proteins, we call that a signal anchor, not a signal sequence, because this signal anchor is actually usually the first transmembrane domain, or transmembrane helix, right? So this is actually going to be embedded in the lipid bilayer. Once that emerges, that signal anchor is recognized by a complex called the signal recognition particle. The signal recognition particle binds to this signal, binds to the ribosome, and takes this whole complex to the endoplasmic reticulum membrane, where it encounters the signal, the SRP receptor. The SRP receptor then works in concert with SRP to release the ribosome, release the new protein as it's being made into the Sec61 complex. So the Sec61 is a protein complex that's responsible for forming a hole to allow proteins to go across, but also opening laterally to allow transmembrane domains to exit into the plane of the bilayer. Now, some of you actually probably remember Shua Shen, a colleague of Doug and myself here at Caltech, gave a Watson lecture talking exclusively about the beautiful work they've done in understanding the mechanistic aspects of this process. This process, because it's so involved in secretion, is called the Sec pathway for short. And in this process, one of the components is that it's co-translational. And so the ribosome, the translational machinery, is still making the protein when the whole system gets engaged, and trafficking across this and into the membrane is done in a co-translational process, resulting in a membrane protein embedded in the bilayer, with each of these being transmembrane helices. Okay, so one final little note about the Sec61 complex. So this was actually a complex that I worked on during my postdoctoral work at Harvard with Tom Rappaport and Steve Harrison. And what was really exciting about this system was that the Sec61 complex is a universal complex for protein secretion and membrane protein insertion. Okay, in all organisms contain a copy of this complex, and most membrane proteins are made by this complex. And while at Harvard, I was part of a team that worked to solve the structure of this complex, and what we learned was that it's composed of 3 proteins. There's 2 proteins, in green is the beta subunit, and yellow is a gamma subunit, and the central part is called the alpha subunit. So all of these helices here are called the alpha subunit. Now, from this perspective, we're looking from the top, from the cytoplasm down in the plane of the membrane. If we rotate that 90 degrees, we get a picture that looks like this. And so now we're looking in the plane of the membrane. These 2 lines would define the lipid bilayer. This is just a cartoon rotating around this. Okay, so what did the structure tell us? Well, the simple thing I can say is that we just look, we know the alpha subunit's responsible for this whole process of putting membrane proteins into the bilayer. And the structure we solved in the cartoon version is a closed form. Somehow, the closed form basically takes the 2 halves of the molecule, so in blue is the first half of this alpha subunit, and in red is the second half of the alpha subunit. They come together like a clamshell. And in the middle is a plug domain. There's a little helix there, which you can see here in the middle, which we call the plug domain, which was responsible for keeping this in a closed conformation. But yet, this closed conformation has to change. It has to open up to form a hole to allow proteins to thread across. The secreted proteins all have to traffic themselves. They all have to travel through this in an unfolded fashion, so that the thread proteins through this. The ribosome has to thread proteins through this. But it also has to be able to open laterally in the plane of the membrane to release these transmembrane helices. And the structure really gave us indication on how you do that, because this clamshell is closed on one end, but it's open on the other end. And so if you move this plug out of the way, you can then make a hole, and even more more so, if you open up the one side of the clamshell, you can allow transmembrane helices to escape into the plane of the bilayer, right? And that would be right here. And so in this orientation, the transmembrane domains would be coming out towards you. And so now, okay, so we've got a really great understanding, at least at a very basic level, about how membrane proteins are, most membrane proteins are made. But the problem is that not all proteins have signals at their N-terminus. Some proteins and so in for the case of those proteins that don't have signals at their N-terminus, what do you do? And these proteins, a classic example of these are classic proteins called tail anchor proteins, and this is one of the focuses of the research in my group. And so tail anchor proteins are different from other membrane proteins in that they have their transmembrane helix, they have a single transmembrane helix at their C-terminus. And so the C-terminus means that it's at the very end of the protein. That's why it's called the tail. And so when translation finishes, that protein is going to be released into the cytoplasm. And when so it's released to cytoplasm, and so it can't actually engage the sec machinery. And so then something else has to happen, right? That's trafficked some other way. Now, tail anchor proteins turn out to be an important class of membrane proteins. In you and me, about 2% of all of our genome encodes for membrane proteins, so it's still a lot of proteins. And in fact, a lot of essential genes are involved in this. And so things like cell death genes that are associated with apoptosis and cancers are tail anchor proteins. And in fact, the sec complex itself, the two smaller proteins, are both tail anchor proteins. These tail anchor proteins have to get to the biological membranes. In eukaryotes, these tail anchor proteins get trafficked in a cell. It's going to go, okay. They get trafficked to a cell, they either go to the ER or mitochondria, right? So sort of two different places where they can go. So already we've got complexity. We've got lots of membranes where these guys could get trafficked to, specifically two that we have to really distinguish between. But we also have to do it in a sense after translation. So this hydrophobic transmembrane domain, which by itself will rapidly aggregate, somehow needs to be protected and delivered to the correct membrane. And so the real question is, how are these proteins, and the talk we're going to do, the rest of the talk is going to be focused on trying to understand how these proteins are delivered directly specifically to the ER. Okay, so the components of our understanding for this really started in the mid-90s, about the time that I was finishing college. And my PhD advisor, Tom Rappaport, was actually the first to comment and appreciate, or my postdoc advisor, sorry, Tom Rappaport, was the first to appreciate that these tail anchor proteins were special class, that they actually had a different problem from most membrane proteins and how they could be targeted. And his lab was the first to demonstrate that they were sec independent. They didn't go via the sec pathway, but they also required an energy source, ATP, to be delivered, which was actually kind of unique already. So from 1995, we didn't actually have any more information about this till 2007, significant information. And that first result came from Manu Hegde's group at the NIH, but also about the same time, Bernard Doverstein's lab in Heidelberg in Germany came up with a similar result. And that was that they identified a protein, which in yeast is called GET3. This protein GET3 is an ATPase, so we satisfy this requirement. So ATPases consume ATP, and that's our energy source for targeting. But also GET3 was known at the time to be a homodimer, meaning that there are two copies of GET3 that come together to form a dimer. And when they discovered this and could show that this GET3 specifically binds to this transmembrane helix, right, this hydrophobic transmembrane helix, that's what grabs it, protects it, and is required for this targeting apparatus. Once they made this connection biochemically, then actually, stepping back, genetics had actually already put together a whole pathway for us. And so what we find is that the pathway was really laid out. There was a clear genetic pathway laid out by people like Jonathan Weissman and Nev Krogan up at UCSF, who developed some really amazing large-scale genetic screens, and they, in that process, identified these other proteins. So the GET pathway, as they termed it, referred to as guided entry of tail anchor proteins, contains 6 proteins. GET3 we know binds tail anchors and is critical for delivery to the ER. But in addition to that, there's GET1 and GET2, GET4, GET5, and another protein complex called SGT2. All right, so that's where the geneticists get to you. But what do you do if you're a structural biologist like myself or a biochemist and you want to understand more details? And so the quest that we had was to take that kind of sort of simple a simple genetic model and try to develop a molecular model for understanding this pathway. And so that's the quest that we set out on. Of course, like any good quest, you need to, you can't just have a leader. You need to have knights who are willing to go on the quest. And so the knights in this case are a bunch of really talented graduate students. And so the graduate students who did this work are here: Christian Soloway, Justin Chartron, Harry Christic, and Amanda Mock. They're a pretty good crew, and so everything I'm going to tell you about in the remaining 15 minutes is going to be related to work that they actually did in the lab. All right, so biochemically, we actually could piece together a much more complex pathway than the one that we just saw genetically. And so this more complex pathway looks like this. Right? And so here we have all the proteins. Now they're arranged in a way that kind of highlights the different major steps that have to occur to get a tail anchor protein to the endoplasmic reticulum. And so we take all of our players, and so we start at the ribosome. Tail anchor protein has to come off the ribosome. We actually don't understand what happens after it comes off the ribosome, but we know that eventually after it coming off the ribosome, it ends up on SGT2. All right, so SGT2 is going to bind, and that's the first committed step to this pathway. Now down this other line here, we've got these other proteins. GET3, eventually we know, is GET3 is going to actually bind the tail anchor and be responsible for trafficking it to the endoplasmic reticulum, where it's going to encounter two proteins, GET1 and GET2. And we don't actually understand mechanistically how this works, but somehow the tail anchor is going to be released into the bilayer here. So the other players in this is a protein complex called GET4 and GET5. It was known prior to us working on this that GET4 and GET5 formed a stable heterodimer, so two different proteins come together to form a stable interaction. And what biochemically our lab and others demonstrated was that GET3 binds to GET4, SGT2 binds the tail anchor, and then GET4 and GET5 bridge the interaction between GET3 and SGT2, where lead to a handoff of this transmembrane domain to GET3, and then that becomes a soluble complex which gets delivered to the ER. And so the story that we're going to relate is how these soluble components work in concert to deliver a tail anchor protein from the ribosome to GET3. And the first place we started as structural biologists, we want to understand the atomic structure of GET3. And so that was a task taken on by Christian Soloway. And so when you want to determine a protein structure, you basically start at the genomic level, right? And so we turn to the genome, and we identified in yeast a fungal GET3 homolog. And so we find that gene in the genome. That gene defines a protein sequence, and the protein sequence looks like this, right? And so the gene encodes for this linear polymer of amino acids. And so what we want to understand is how this linear polymer of amino acids is assembled together in a three-dimensional structure, and that's what we really do as structural biologists. So that's great. We understand that this gene structure gives us protein structure, but we can't just make this ourselves. We actually have to turn to bacteria, and so we're actually going to engineer bacteria to make this protein for us. And so when we engineer that bacteria to make this protein for us, when we do this in E. coli will mass produce this protein. We will then have to take that protein out of E. coli, purify it, and get it to a sample that's capable of forming crystals. Right? We're X-ray crystallographers. As structural biologists. And so that process starts with crystals. Once you have crystals, so these are crystals of GET3. Once you have these crystals, you can then put them in an X-ray beam. And that X-ray beam is going to hit those crystals. And where we do this is up at Stanford. The Stanford Synchrotron Radiation Laboratory in Palo Alto is a large synchrotron. It's this huge machine. And the purpose of that machine is to generate high-energy X-rays. And Caltech, through the gifts of Gordon Betty Moore Foundation, actually built a beamline up there that's one of the best beamlines in the world. And so we're really lucky here at Caltech to have access to that. And so what we do is we take our crystals up to Stanford and we shoot them with these X-ray beam, and then we get a diffraction pattern, which is a bunch of spots. We can take those spots and computationally convert them into a molecular structure. And that molecular structure that we get really defines the location of the electrons. So we can see this map that defines the electrons. But because this map is is a three-dimensional map, and we know what our protein sequence is, we know the structure of amino acids and how they're linked together in a polymer, we can take this map and build an atomic model into it. And so that's the structure that we get. And so for GET3, we went through this whole entire process and eventually solved the structure that looks like this. And this is the structure of GET3. As I said, it's a homodimer, right? And you look at it from this perspective, we're really just highlighting all of the atoms. It's sort of hard to get any appreciation for what it's doing here. And so let's remove some of this information to make it a little easier to look at. And so here I'm just converting it into what we call a cartoon model. And what we have in this cartoon model, each of these lines and these shapes are actually following the backbone of the amino acids. And so what we find is that we're just tracing through that, and we get a structure like this. And what you can see, I've colored it in a way, so now you can see there's two copies of this molecule. So there's a symmetry axis down the middle here. So there's a left subunit and a right subunit. Bound in our structure is actually is an ADP molecule. So it's like ATP, except it's missing a phosphate, and that's those are ADP there. So each of these subunits binds to an ADP. And because it's bound to an ADP, the structure that we solved is what we now know, and at the time predicted, was an open structure of GET3. And so here we have these two molecules bound to ADP in an open conformation. And what we predicted based on this on the structure was that if we actually add another phosphate here, that's going to have dramatic repercussions where this molecule is going to reorient itself. And that reorientation is going to drive these interfaces together, bringing these nucleotides actually closer. The other feature that sort of stands out in the GET3 molecule are these loops that kind of stick out the top here, right? These kind of rabbit ears that come off the top. And what was really interesting at the time when we solved the structure was that these loops contain a lot of hydrophobic residues that are exposed. And that's unusual for molecules. And so we rationalized at the time that the binding to ATP would lead to a rotation, and that these loops would reorganize themselves into a way where they'd be capable of binding to this hydrophobic transmembrane helix of the tail anchor. So subsequent work from our lab and lots of other labs actually produced a series of structures, and all of them really corroborated our model, which was that we have an open structure, we have a dimer that's an open structure. Binding to ATP drives or rotation across the front. So this is an overlay of a bunch of different crystal structures, but they all point to the same story, where you have an open conformation and a closed conformation. And that rotation across the interface happens exactly as we predicted, and the result is that you get a closed conformation, that now these loops have rearranged themselves. And so now we're going to redraw this structure on the right as a surface representation. And the reason to do it this way is that we can actually then look at sort of what a molecule would see as it encounters GET3. And I'm going to color it in a way where we've colored these sort of acidic residues in red, basic residues in blue, and then we're now going to in the hydrophobic residues in green. The hydrophobic residues are going to be the ones that are going to interact with our tail anchor protein. And so we just do a movie where we now rotate around that. What we find is that a groove had been formed now across the top. That's a hydrophobic groove, and we can then predict, and this was actually first predicted, the first structure that gave us insight into this came from actually a colleague of mine at University of Chicago, Bob Keenan's group. And when they saw the structure, they actually made the prediction, which seemed reasonable at the time, that a alpha helix would fit nicely into this groove, and that's how GET3 binds. And so we can start building up a more complex picture now when we go back to our cartoon here, where we can replace these pieces with molecular structures. And so we can do this, so now we can do this for each one of these steps, right? So GET3, we saw the structure of GET3, we understand how that works, or what that looks like. And so then the next thing we became interested in is this GET4 and GET5 complex. And one of the things we determined fairly early on was that GET4 and GET5 don't actually behave aren't actually just a simple heterodimer. They're actually heterotetramer, where GET5 dimerizes. And so two GET4s, each GET4 is bound to a single GET5, and then GET5 dimerizes by itself. We saw structures of components of this to bring together a molecular model, where we can now fill this cartoon in to get a structure that looks like this. And so here's our dimerization domain. There's a central domain for GET5, and then here's the structure of GET4. Now, we knew biochemically that GET4 is responsible for binding to GET3, and so we wanted to actually understand this structurally, and so we next went on to try and get some picture view of what this complex looks like. And so Harry Gristick and the group went on to solve the structure of this GET4 bound to GET3. And so we can now add that into our molecular model here. And what we see is a structure like this, where a dimer of GET3. Now, what we knew going into this was that the GET3 or GET4 was going to recognize a closed form of GET3 in a specific way. And so the structure we solved was of a closed form of GET3, or Harry solved the structure of a closed GET3 bound to GET4. What we knew biochemically, and this is in collaboration with Shuo Shan's group, was that GET4 actually not only does it prefer a closed form, but actually prevents hydrolysis of ATP. And that's important because you need to maintain this orientation for delivery of a tail anchor protein. So the next place that we went to look was to try to understand the structure of SGT2. SGT2 is the first place where this complex comes into where the tail anchor protein encounters the complex. And like the GET4-5 complex, it's a multi-domain protein. It's got a dimerization domain. It's got the central domain that actually interacts with a class of chaperones. And then it's got this N-terminal domain whose structure we're still missing. And we know this N-terminal domain binds to tail anchor proteins, but we're actually missing atomic resolution. So this we're working very hard on. But the molecular architecture of SGT2 looks like something like this. So now we want to fill in this next part of the piece of the puzzle here. So the next thing we went on to solve was a structure of how SGT2 interacts with this complex. We knew SGT2 binds to the central domain of GET5, and so the dimerization domain of SGT2 binds to the central domain of GET5, and so we went on to solve a structure of that. It looks like this. Now that we have this structure, we now can piece these structures together in a very precise way, and so then now adding all of these structures gets us the architecture of what something like this would look like. And so now we can replace, all of these. Now I can replace this cartoon with a picture that looks like this. Okay, so the final thing now in this puzzle is how does GET3 look when it actually binds a tail anchor? And so, what I'm going to tell you is we don't actually have a structure of that yet, and so we're still working very hard on it. But we do have some, we think, molecular insights into what's going on here. And one of these was the fact that when we purified GET3, it had some weird behavior. And one of the things is that if you took GET3 and you ran it on what we call a gel filtration column, which separates molecules based on size, smaller molecules run faster, and using UV absorbance, we basically can trace molecules they come across, and so we can get a line like this. And so this is just watching liquid go past a UV detector, and the more protein, the higher this goes. And most of this, the protein in this is GET3, and so what we could see is that there was a dimer peak for GET3, and then a larger peak that represented a tetramer peak of GET3. And that was surprising. If you look back at that structure, as we said, there's a dimer, not a tetramer, and yet we could take this tetramer peak and repurify it. Now, okay, we didn't really understand what was going on there, but we did know that we could purify complexes of GET3 bound to tail anchor, and so in doing so, we could take those complexes and do a similar experiment as we see here, and we took those tail anchor complexes of GET3 bound to tail anchor. What we found was that those tail anchor complexes completely localized to this tetramer, right? And so at least in this system, the tail anchor protein is binding to a tetramer, not to a dimer. So that suggests that our molecular model is wrong. So as I said, we don't actually have a structure of the yeast tetramer, but we do have insight molecularly to it because we turn to another kingdom of life, archaea. And so it turns out archaea, the branch that archaea is on, actually includes species that contain GET3 homologs. Now, unlike eukaryotes, we have all these organelles, archaea look more like this. No organelles, and so why do you have a targeting factor like GET3 in archaea? We don't know the answer to that. What I can tell you is that these proteins behave very similar to the fungal system, and so we think that this is a good model system for that. And of course, the thing you do then is you go to solve structures, and so we took one of these archaeal homologs. We actually knew at the time that these archaeal homologs actually purified as tetramers, and so we had a good hope that this would actually give us some insight into the tetramer structure. We solve the structure of one of these, and sure enough, we get a tetramer of GET3. Now, here we have two dimers juxtaposed relative to each other, and so instead of having dimers with grooves, we have a single dimer with a groove. Those grooves come together, they interdigitate, and the result is that they form a complex where if we now do a surface representation where we've cut through the middle, but trying to get the cartoon similar to what we saw a minute ago, what we get is something that looks like this, where the resulting surface representation shows that inside here, instead of having a hydrophobic groove, we have a hydrophobic chamber. We've put an alpha helix of the appropriate size. What we now believe is that what's happening is that there's a transmembrane domain trapped inside this hydrophobic chamber, and so the tail anchor would trap this domain. And so now we can build in at least what we think molecularly is happening, and we have lots of evidence for why this is probably a good model. And so now to fill this in, we know that we believe that GET3 probably doesn't traffic as a dimer, it actually traffics as a tetramer, and we can replace now this with a tetramer structure to get something that looks like this. All right, and so the result is that we basically now have defined this whole pathway at the molecular level. All right, and so to wrap up, we can basically take this molecular pathway where we've solved all of these structures, and there's lots of mechanistic insight that I'm obviously not going into, but I'd obviously be happy to talk about if people are interested. But we can now take that and make it a sort of a simpler cartoon where we know that tail anchor proteins come off the ribosome. Now, we don't know what happens next, but we do believe that there are molecules called chaperones that may be involved in this process. And so these chaperones might be responsible for delivering tail anchors to SGT2, but they don't have to be. But if they are, we still need to work that out. But that delivery of these tail anchor proteins to SGT2 then binds them. SGT2 then will interact with GET5, And so SGT2 will interact with Get5, it will bind Get5. We actually demonstrate that this interaction is rapid kinetically. And so this SGT2 is rapidly sampling Get5. And when a tail anchor is delivered, eventually that tail anchor will get transferred into Get4, into Get3. Get3 will then reassemble to actually form this closed tetramer complex. The consequence of that, which I kind of skipped over, is the fact that when the Get4 holds us in an orientation where we have this open conformation, where ATP can't be hydrolyzed. We think tail anchor binding actually will drive ATP hydrolysis, resulting in that tetramer structure that will release this from the rest of the proteins, and now we can get traffic to the ER for insertion. So that's what happens in yeast, okay? And that's interesting for a lot of important biological reasons, but what we really care about is what's happening in metazoans, okay? Apes, which, as my son likes to talk about, includes us. And so what's happening in us? It turns out that in us, this process is actually linked to a surveillance system in the cytosol for recognizing hydrophobic domains. And that makes sense because tail anchor proteins are hydrophobic domains that are localized to the cytoplasm. But unlike this pathway here, we actually have all the pieces of the pathway here, so you can just map the colors. I kept the colors the same to kind of map the players. And we have all these players here, except that in addition to these players, we've actually added now this protein called BAG6. And what BAG6 does is instead of having a dimeric interaction of Get5, that interaction is replaced by BAG6, where it binds both TRC35, which is the Get4 homolog, and UBL4A, which is the Get5 homolog. That makes a heterotrimeric complex. And what happens in this case is exactly what we saw before. We get this trafficking of tail anchors eventually to the Get3 homolog, which gets delivered to the ER. However, we also then this complex is responsible for recognizing membrane proteins that are in the cytoplasm that shouldn't be there. And when it recognizes those, it will grab onto them by SGT2, deliver them to BAG6, and then tag them for degradation. So we'll say it will flag these for the trash. And so we actually recently, just this week, published a paper which really gives us the first molecular understanding of this complex. And so this is where we're really interested in going and trying to understand exactly the interplays that are happening in us and how these might be linked to a lot of diseases. All right, so finally, why should we care, right? I mean, membrane protein biogenesis is interesting to academics, but why should it be important for science? And I think, one sort of simple answer is that, current evidence suggests that a lot of diseases have their roots in membrane protein biogenesis. And in fact, cystic fibrosis is one of the most famous cases of this, but there's lots of other diseases as well. And I think as we begin to understand many disease mechanisms in more detail, certainly a lot of cancers, we'll begin to understand that these processes are really important to understand. In addition, understanding membrane protein biogenesis is great because membrane proteins do a lot of amazing things. And so if we're trying to engineer biological systems, being able to do that properly is going to require that we understand biogenesis at a level at which we can control it. And so we need to understand how nature does it to be able to engineer it. And I think finally, and for me, I think the most important reason is that basic science is just cool, right? We basically start with a molecular mystery where we don't know what's going on, and through hard work and effort, we actually can take these very opaque pictures and reduce them into molecular understanding. I think there's just nothing that's more exciting in life. I'm a little biased in that, but that's just really amazing. And I think what's really great is that nature, because of the complexity of nature, it just has these limitless quests that are out there for people to pursue. And so for curious people, there's just so many interesting things to do. And I'd also like to point out the fact that basic science is today's basic science is tomorrow's translational research, because the medicines that we have today are all rooted in basic science, and it's understanding these mechanisms which allows us to make predictions about the future. All right, so in light of time, I'll just quickly say, my lab has a lot of amazing and talented people, and our work is, I've already mentioned the grad students, but I would like to add that, this has been a very, happy collaboration with my colleague here, Shio Shan. 2 students in her lab, Mike Rome and Mira Rao, have really been instrumental players on our project. On this project as well, on the mammalian side, we collaborate with Yihan Yi at the NIH, an old friend, but this has been really beneficial to us. And the final thing I'd like to say, in addition to funding sources, which have been useful, is the fact that we have 2 really great facilities here, a molecular observatory, which allowed us to do all of our crystallography and just makes Caltech one of the best places in the world to be, or maybe the best place in the world to be a structural biologist. But also we have an NMR facility here that's supported by a really great scientist, David Vandervelde. And I didn't admit to this, but some of the structures we solve were NMR structures. And so we couldn't have done that without that resource here as well. And so I'm happy to take questions at the front, so if you have any questions or discussion you'd like to have, feel free to come on up and chat. Thanks for your time.